লাইভটা কি চলছে? তাহলে শুরু করে দিই নাকি? হ্যাঁ, আরে আবুদাকে মোবাইলটা একটু ইয়ে করতে রাখো না।
হ্যাঁ, দেখো তো ইউটিউবটা চেক করো তো। হ্যাঁ হ্যাঁ, শুরু করো। আমার একটু প্রবলেম হবে, নেটওয়ার্ক একটু স্লো। আমি দেবুদাকে আবার হোস্ট করে দিচ্ছি। দেবুদাকে শুরু করতে বলবো।
So I hope it is on track now. On behalf of the organizing committee, I really pardon from you due to our inaugural session is not being lived in the YouTube. But as the technical difficulty, as we have promised that from the first technical session it will be on. So let's move on to the first technical session where Dr. Nirmalu Dasgupta will deliver his first lecture. So let me introduce Dr. Dasgupta. Uh, Dr. Dasgupta completed his BSc and MSc from University of Calcutta and PhD from biochemistry from the National Institute of Cholera and Enteric Diseases, NICED, ICMR, and University of Calcutta. He has been awarded several fellowships like Glenn Foundation for Medical Research Postdoctoral Fellowship in Aging Research, ICMR Senior Research Fellowship, University Grants Commission Junior Research Fellowship. Several research experience and employment are as follows. He is now the postdoctoral fellow and principal investigator at Sanford Burnham Previs Medical Discovery Institute, San Diego, California, USA. And he was associated, as I mentioned, in the NICED ICMR. He is now working on the aging research. He has several research peer-reviewed publications in his name. And with this brief introduction, I now request Dr. Dasgupta, who is also the son of the soil, means where our college is situated. He is also from Belgorия. So with this short introduction, I will request Dr. Dasgupta to deliver his first lecture. Over to you, Dr. Dasgupta.
Okay, thank you. Am I audible?
Yes, you're perfectly audible.
Okay, thank you. Thank you, Dr. Bhadra, for your kind words. And I would like to thank you and other organizers for inviting me in this international webinar on COVID-19. And it's really a great pleasure because, as you told, I am a son of soil. So Vairab Ganguly is my Parar college. And another reason for that, because today I am sharing the virtual platform with my professor, my teacher, who taught us the first virology in our MSc classes, the keynote speaker of this webinar, Professor Dhrubajyoti Chattopadhyay. It's a really surprise and great pleasure for me. Honorable Vice Chancellor is not present now, so I would address other distinguished faculty members and Vice Principal, Honorable Keynote Speaker, and other respected guests, and my dear students. Before I start, I want to share my screen. Dr. Bhadra, please tell me that if my screen is visible or not. Is it visible, Dr. Bhadra?
Yes, yes.
Okay, thank you. Because I'm the first speaker of this webinar, so I would try to introduce SARS-CoV-2 and COVID-19. I will touch different aspects briefly. I think that next speakers who will cover it more extensively and in depth. So SARS-CoV-2 is a novel coronavirus. The name is Severe Acute Respiratory Syndrome, Coronavirus 2. Why it's 2? Why not 1? I will come later. And the disease caused by the SARS-CoV-2, that is COVID-19. That means coronavirus infected disease in 2019. So the name of the virus is SARS-CoV-2, and the disease caused by SARS-CoV-2 is COVID-19. I divided my talk in 4 segments: that what is SARS-CoV-2, and where did it come from, what is the general information known about COVID-19 and SARS-CoV-2, and what are the different approaches are being developed for the vaccine and drugs. So these are the 4 segments. Before entering into the coronavirus, because there are a lot of students who are mostly from physics background, I want to tell few sentences about virus. That what is virus? The first question is that are viruses alive? We can't say it's viruses are alive, because virus doesn't have any cell, it doesn't have any active metabolism, it doesn't produce ATP. So we can't say virus is alive. Then can you say it's a non-living? But the argument should be that then virus somehow it reproduce. So you can't say it's non-living. But for virus reproduction, it need a living host cell. Without a host cell, it can't replicate. So after a long debate, now somehow there is a consensus that it is an intermediate stage between the living and non-living. So what is the definition of virus? That viruses are obligate intracellular parasites. That means it needs some host cell to multiply. And what is the genetic material of viruses? From bacteria is a living organism. So from bacteria to human, the genetic material is DNA, and it's double-stranded DNA. For virus, it can be DNA, it can be RNA, and it can be single strand and can be double-stranded. You can follow my laser pointer. This is a double-stranded DNA, and this is the single-stranded DNA. So virus can have double-stranded DNA, double-stranded RNA, or single-stranded DNA, single-stranded RNA. So virus is an assembly of protein, nucleic acids, lipids, and carbohydrates. If you see that this is the genetic material of the viruses, and this is proteins, and most of the viruses, they have one lipid layer, which is called envelope, and few viruses which do not have this lipid layer, they are called naked virus. You can see several viruses, which is you heard about this, like rabies, herpes, pox virus, HIV, and this is the coronavirus. This is the deadly Ebola virus. Now coronavirus. Coronavirus has that envelope, so it is an envelope virus, and its genetic material is RNA, single-stranded RNA. So coronaviruses are a large family of envelope single-stranded RNA virus. And it is divided in 4 groups: alpha, beta, gamma, delta. And the this SARS-CoV-2 and other 2 viruses, what Professor Basho Chaudhary told about, that's MERS and SARS, they are also in the beta coronavirus group. So this SARS-CoV-2 is not the first virus which caused that much morbidity and mortality. It came before the coronavirus outbreak happened before of that. Like in 2002, there's a first outbreak that is called that severe acute respiratory syndrome. That's why it's 1. And the death rate is very high, like 10%. And then in 2013, it is Middle East respiratory syndrome, MERS-CoV-2. The death rate is far higher than 36%. Though the infection rate and spreading is less, so those didn't cause the pandemic. But for this SARS-CoV-2, the spreading is very high, and so it now become the global pandemic. And you can see today it's more than 8.6 million people are infected, and 100 people died. So what is, why we call it as a coronavirus? Because in 1968, the scientists looked under the scope, electron microscope. They found the uh, shape of this virus is like a solar corona. You can see today, because today is a solar eclipse. So this is a, like a crown, this solar corona. Because of this appearance, they named it as a coronavirus. Now we know that this corona is actually the spike protein. Uh, and this spike protein is very important for virus, because with this spike protein, they anchored with our ACE2 receptor, which is angiotensin converting enzyme 2. They anchored, they bind to the ACE2 receptor and fuse with the host membrane, and then enter to the cell. And after entering to our cell, they use their own RNA polymerase to replicate their RNA. And using the host machinery, they produce their structural proteins, and they assemble in our cell, and then a new virus is generated, and it infect other neighboring cells. Now, what's the origin of SARS-CoV-2? It is known that the bats are reservoir of many deadly viruses. And by the previous knowledge, people know that the SARS-CoV-1 and MERS-CoV both are originated from bat, where for SARS-CoV-1, the intermediate was civet cat, in Bengali that's called bhamberal, and for MERS-CoV, it was camel. So from bat through civet cat and camel, it infect human. So that's why scientists first look at the genome sequence of SARS-CoV-2 and the bat coronavirus. And they found that this is horseshoe bat. It is horseshoe because its mouth is like a horseshoe. That's why it's called horseshoe bat. And scientists found that the genome sequence of this SARS-CoV-2 is 96% similar with the horseshoe bat coronavirus. So scientists conclude that it again came from the virus. The origin is bat. Uh, and then what is the intermediate host? Then they found the pangolin coronavirus, the RBD of the pangolin coronavirus. RBD means the receptor binding domain, the domain which of spike protein, which is actually bind to the ACE2 receptor, that is highly similar. It's 97% similar with pangolin coronavirus. So then scientists concluded that it actually originated from bat, and pangolin was the intermediate host. From them, it came to human. Now, how do coronaviruses get into humans? Because in the Wuhan seafood market, somehow these pangolins or maybe some other hosts, they are infected, and they maintain this wet market is very unhygienically, and so it is spreaded in them. And somehow who handle this pangolin, he or she, I don't know, that person got infected, and who consumed that meat of pangolin, they got infected, and that the reason for this outbreak. That outbreak started like this, to handling this meat, pangolin meat. Now the thing is that if you see the paper in 2013, 7 years ago, they mentioned that this coronavirus infection will come again, and it possibly escalate. And we are in that, after 7 years, we can see that it is escalate, and it kill almost 500,000 people. And why is the reason? Because in the last, they also wrote that last 100 years, our population growth is so high that too much encroachment of the wildlife habitat and deforestation. So if you look at this left-hand figure, there is no intersection between the bat territory and human territory. But now bat needs their shelter, bats need food. So they are also coming to human territory, and human territory also increased, and their domestic animal territory, that is also increased. So there is a large intersection of these three. So maybe the SARS-CoV-2 is not the last. Maybe after a few years we can see SARS-CoV-3. It is also possible. And bat can carry many deadly viruses, and like the Ebola, Nipah, Hendra, SARS all came from bat. Uh, and that is a very interesting question, that how do bats carry so many deadly viruses? Uh, that is actually a different topic and can be used as a separate talk. So if you are interested, I will give a talk in Ashutosh College on that. A date has not been fixed yet. That's my fault, because I didn't tell him when I can deliver that, and I wrote one article on that in Desh Patrika on 2nd May 2020 edition. That is, I wrote it. I tried to write it in very lucid manner for everyone. So if you are interested, you can read that, or you can attend that seminar. Now, I don't want to demonize bats, because bats are our friend, not our enemy. And they are the natural pest control. They can consume millions of bugs at a night, and they can help in pollinate plants, and they disperse the seed so rainforest can grow. Uh, and bats, we can't say that bats are the only reservoir. We don't know which wildlife is also reservoir some other deadly viruses. So we have to reduce deforestation. We have to reduce our encroachment to their habitat. That's why we can save us. Demonizing bat is not the solution, and killing the bat is not the solution. Now I will go for COVID-19 timeline. COVID-19 first case was reported on December 21st, and then virus identified on January 7 within 2 weeks, and the genome sequence was available in January 10 in the for everyone. And in the end of January, WHO declares a public health emergency of international concern, and the disease was named COVID-19 on February 11, and March 11, WHO declares it as a pandemic. And now, after more than 3 months, 8.6 million people are already infected, and death is almost now reaching 500,000. There is an interesting thing that from many corners, we are hearing from last 6 months that COVID-19 is nothing, it's just a flu. Even the President of America, he tried to convince people that it's just a flu. So if we consider it's just a flu, if we compare the seasonal flu and COVID-19, first we have to consider the R0 number. R0 number means how many people will be infected by an individual with this disease. Means if one person is infected, he can infect another 1.3%. That's for flu. But for COVID-19, it's 1.5 to 3.5. If you look at this figure, that if for seasonal flu, 5 people is infected, then they can spread it 45 people after 5 cycles. But for COVID-19, if 5 person are infected, after 5 cycles, 368 people are getting infected. And if you don't stop the spreading, it can be a pandemic. You can, and it happen. You can see that result. So R0 number is very high for COVID-19. And if you compare the incubation time for flu, it can be 1 to 4 days. But for COVID-19, it is 1 to 14 days, and maybe 21 days. I heard about that 1 month people are suffering from COVID-19. And because the hospitalization rate is 10-fold higher than the seasonal flu, so you need hospital bed. And if 14 days someone is in the hospital, other people can't get the proper treatment. And it happened in New York and Italy that people didn't have bed. The hospital system was collapsed, and people died for that without treatment. And the fatality rate is 0.1% or less. For COVID-19, now the pandemic is going on, and because of the asymptomatic carrier, we don't know what is the actual rate, but it is estimated it's close to 1%. So from this comparison, we can't say that the COVID-19 is just a flu. It's not just a flu. We should be more focused, should be more concerned about this COVID-19. Now, if we think that how deadly is SARS-CoV-2, if you look at the x-axis, it is people infected by each sick person, means higher means the infectivity rate is higher, and this y-axis is fatality rate. You can see the Ebola, where the fatality rate was 50%, and the bird flu, smallpox, Spanish flu, the infectivity is very high, but the spreading was less. But if you see the measles, chickenpox, the mortality is very less, though they are highly contagious. We don't have a virus here. If we have a virus here, then the maybe world population will be died within few months. But it is not possible because it is against the virus evolution. Virus needs some host. If it kills the host, then it can't propagate and it can't multiply. So you can see the deadly viruses, infectivity of the deadly viruses is less. Now this SARS-CoV-2, because still the pandemic is going on, we don't know properly the number. You can see the region where it is in between of all. Like it is the spreading rate is like 3.5, and the death rate is like a median of 1%. Now there is another controversy and conspiracy theory that it's a lab made. So I would tell that it is very difficult to make it lab, which is all this property that which can spread fast and also kill many patients. So to maintain this mid region, I don't think it's possible. So it's a scientist also several times wrote that it's not a lab-made virus. It has its natural origin. It came from bat through pangolin. It infected human, and now it's a human virus. There is a very interesting paper came last week that it is a small population, but I think it is very important to know because the epidemiologist estimation is supported by this data. If you see that there is a cruise which was going to Antarctica. They started their voyage on mid-March, and there were this many passengers and crew, like 2027, and nobody had no symptoms on COVID-19. But after a few days, one patient showed a symptom, and eventually the 60%, 59% of the passengers and crew got infected with the COVID-19. And surprisingly, this 81% of these 128 people are asymptomatic, which is always being talked that the more are the asymptomatic than the symptomatic, and 19% was symptomatic. And among this 19%, 6% need hospitalization, 50% of them need ventilation, and 1% died. That means almost 1%. So this estimation, I think it's very close to the estimation which are already discussed several times, that 80% people are asymptomatic. So we don't know that who are asymptomatic. So we need to be careful, because you can say that the death rate is very high in old people, but there is a 20% overall hospitalization rate for younger people too. And if we don't have enough bed, that can be fatal, because maybe you need some oxygen, but you can't give that oxygen. That's why a person can die. Now, I will tell about the vaccines, what the different approaches are going on. Most of the people, most of the lab or industry, they are trying to develop a vaccine to use S protein as an antigen. And there are different approaches. like RNA vaccine, where the S protein RNA will be delivered, where it produces the S protein and build the immunity against SARS-CoV-2. Similar like DNA vaccine, where DNA, RNA, and then protein. And some groups, they are trying to inject the recombinant protein vaccines, the S protein, which will develop the adaptive immunity against the SARS-CoV-2. And this is the famous because Oxford vector vaccine, the Chadox vaccine, the adenovirus, which is a different virus, which is a virulent strain, and they integrated the S protein genome into that adenovirus, and they will inject this, and then after its infection, S protein will be generated and produce the adaptive immunity against the SARS-CoV-2. And two straightforward approaches: inactivated vaccine and live attenuated vaccine. Inactivated vaccine means that producing the SARS-CoV-2 in a laboratory by in cell culture, and then inactivate it by chemical measure. For now, it is using the beta-propiolactone, so that virus can't replicate in our cell, but it can give the immunity against SARS-CoV-2. And live attenuated vaccine is, it is a virulent strain that growing it in the culture, when it become virulent, it become less weak, it become weak, then it can be used as the vaccine. So if you see that there are 135 clinical trial is going on, and now 125 in the preclinical stage, 7 is phase 1, 7 are in phase 2, and 2 are in phase 3, and still now we don't have any approval for the actual vaccine. So all this platform has their own advantages and disadvantages. Like for RNA vaccine and DNA vaccines, the production is very fast. It can be produced rapidly. But the thing is, this platform is not used earlier, so we don't know the safety. So safety measures should be taken, and that would take time. For recombinant S vaccine, it is slow, but it is already established. It was used, these approaches. But for a global level production, it need time. For viral vector-based vaccine, which is by Oxford, University of Oxford and other industry and institute, that is also some advantages of that it is not using any infectious virus, but it can happen that the virus actually being used, that can affect somehow the immunity. It is possible. So scientists have to look at that. And there are two straightforward approaches, like live attenuated vaccine and inactivated vaccine. The problem with this is that we need time to make the virus when it becomes virulent. So it will take time. You are growing, and you have to take a lot of time for the extensive safety measures. And for inactivated vaccine, the disadvantage is that you have to culture a lot of infectious virus particle, and that's the disadvantage of this inactivated vaccine approach. But both are approaches are very straightforward. And one thing I should mention, that Serum Institute of India, they are also trying to develop the vaccine. They are using that live attenuated vaccine approach. So it would be very good that if we have that vaccine from our own country for us, it would be cheaper. Let's hope for the best, and we should wish Serum Institute their success. Now the drug targets. I would not mention the other drug targets because those are already failed. So it's a wastage of time to discuss about those. Two type of drugs now are in clinical trial. One is that TMPRSS2. This is the protein which facilitates the binding of ACE2 receptor and S protein. It is a serine protease. So the clinical trial is going on against this protein, the inhibitors, and maybe it will take, I saw the clinicaltrial.gov.in, they are mentioning it will take 2 years from now to conclude. And another is that the inhibitor of the RNA-dependent RNA polymerase, the viral RNA polymerase. I think that you all heard about that remdesivir. There are 2 clinical trials, but this result is confusing. One group, they reported that there is no advantage using remdesivir. Another group, they are telling that it reduces the hospitalization period, like 14 days to 11 days. But I think we can't conclude right now. We have to wait for more number of the patients and the result. So these 2 approaches are going on. Now you can ask that what are we doing? Means our institute. First, I need to clarify that I am not in this group, but I am presenting it as an institute person, as an employee. You saw that the production of a vaccine could take 12 to 18 months or more, because there is phase 1, phase 2, phase 3 clinical trials. Then you need license, you need to register it for global production, administration. It will take time. And for new, if you start from a new drug, you design a new drug, you synthesize that for safety, efficacy, any new drug, they usually requires 10 to 17 years. So we can't wait for that. So what we are doing, we are repositioning the clinically evaluated drugs. Means the drugs which are already FDA approved, or it is using in clinical trials or preclinical trials for other diseases. We are testing these 13,000 drugs. We have already screened those drugs. What is the approach? That we infected the green monkey kidney cell, VERO-E6 cell, with these viruses, and we saw this is called the cytopathic effect. If virus replicate within the cell, it kill the cells, which is called cytopathic effect. And we measure the cytopathic effect in presence of this compound. That if this compound, some of those inhibit the virus replication, cell would not die, it will be alive. So we measure the ATP of these cells, because ATP is the marker of that cells are alive, and we screen very fast these 30,000 compounds. And we found that 30 molecules which have these antiviral activities against SARS-CoV-2, and four of them are FDA approved. One drug is registered in Japan, and 11 molecules have in the preclinical stage and different clinical trial stage. And we also received 10 million grant support from government of USA for testing the broad spectrum antiviral to combat COVID-19. Though it was not, this grant was not written for COVID-19, that department sent the proposal earlier than the COVID, but now government is telling us to do the study for COVID-19. Now lockdown is over, and we are listening the term herd immunity. Herd means a large population of animals. That means herd. The concept of the herd immunity is that if a large population is infected or vaccinated, so the rest of the population is protected from this virus, because the infected or vaccinated people, they stop the chain of the infection, so rest of the population is also protected. So virus is disappeared from the environment eventually. But there is a new paper, according to their estimation, that you need at least 67% of people get infected and from the SARS-CoV-2, then you can establish the herd immunity. So where we stand now, if you see that the New York City, a number of death and infection you heard about, but see, still it is only 20%. the infection. London, it is also 17%. Sweden, the Sweden was advocating for the herd immunity from the day one, but still in Stockholm, it's only 7.3% infection. And Wuhan, the epicenter of the SARS-CoV-2, still there is only 10% people are infected. So we are far from the herd immunity. And if you consider the stat, still only for today's point of view, it is only 0.11% of the world population is infected. And if we need herd immunity, we have to infect it's 5 billion people, which is we are far from that. So I would tell that herd immunity is very difficult for now. We don't have vaccine. We don't have any drug. So just take precautions, protect yourself and other from COVID-19, and clean your hands often, avoid close contacts, stay home if you are sick, cover cough and sneeze, and wear face mask. Face mask will really help you to, not only you, it will protect your other person from you. So everyone should use the mask and stay home. I know it's not possible because I have to also go to my institute, but reduce the as possible as you can the going outside. So that is all from me. Thank you. This is one of the building of our institute. I would like to thank again to the Devabroto and other organizers. And now I am ready to take the questions. Am I audible? Am I audible? So if so if there is no yeah, Devabroto, I am ready for any question.
Thank you very much, Dr. Dasgupta,
Yes,
for
Dr.
your
Dasgupta is coming. Okay.
illuminating lecture. We can entertain the 5 questions because our next panelist is already on. So from the Zoom, if anyone is interested, kindly unmute and question Dr. Dasgupta.
And you can ask me in Bengali also. There is no problem.
One of our participant, Dr. Soili Biswas Adhikari.
Yes.
Dr. Dasgupta, am I audible to you?
Yes,
Yeah,
yes.
sure. Please go on.
Dr. Soili Biswas Adhikari, he has asked some question to you, sir. When will vaccine come, sir?
When according to I think it will take another at least 1 year to 2 years. And you know that vaccine, most of the vaccine is being failed at the phase 3. So I hope that vaccine, this S protein, it will work and the virus will not be mutated that much that this vaccine will not work, but it will still take time. I know that many companies and the institute, they are claiming that they are very soon they will deliver the vaccine, but I don't think that's possible. I talk many people, the virologists and the vaccine persons in our institute, so they They even personally, they are telling that maybe it will take 3 years.
Another participant, Indranil Bhattacharya, he is asking a question to you, sir. You have said that we wear masks if you are sick.
Your voice is broken now.
Am I audible, Nirmaloda?
Yeah, you are audible.
So the next question is from Sandeep. Will herd immunity will work for India?
As I told that you need at least 60% of the population getting infected or getting vaccinated. Without vaccine, without drug, it will be very risky affair, because if 60% population need to be infected, the hospital system will be collapsed. And I don't know. It's very tricky question, and we don't have any answer for that, because now only still 0.11% people are infected.
So the next question is from Ritik Sarkar. How much helpful are anti-inflammatory drugs? Can it really help to reduce the load on the health system?
Actually, virus doesn't kill us. It is told our immune system kills us when the inflammation is too high. So doctor use that immune suppressant. Like that BBC is now heavily mentioning about that dexamethasone, which is not a new thing, but they are claiming because it's I think because of the research from UK. So they are like it's a miraculous drug. It's not a miraculous drug. People use immune suppressant, the steroid and IL-6 receptor inhibitors for treatment, but it is when the immune response is very high, you need ventilation, you need oxygen, that time that if you are taking any immune suppressant, it is not good for you, because your your immune system will clear the viral load by your own immune system.
Okay, the next, there are many questions, but we cannot entertain the silly
Yeah,
ones.
my professor and my sir will be there. He can also answer.
So the next question, if a person have no symptom and COVID positive, what will we do with them?
Means asymptomatic.
Yeah, yeah.
We don't know, because even the asymptomatic patient, we don't know, means like he already had that disease. Now if you doing the qPCR, you will not get the virus, you can't detect the virus. But if you detect the antibody of IgG or IgM, IgA, then you can tell that this person was infected and now he is immune. Probably he is immune. We don't have enough evidence that we can say that once infection that is make you immune, but theoretically it would be.
Okay, the next question is from the zoology professor of our college, Dr. Saurabh Som. Where does coronavirus stand in David Baltimore's classification scheme of viruses?
I really don't know. My sir can tell this.
So the next question, apart from maintaining hand hygiene, what else can people do to protect themselves?
Wear face mask, because initially who told us that face mask will not work, but it is now reported for everywhere that face mask will work. You can see the report that from Italy that some doctors, they are claiming that this virus is different than 2 months before, but it is not like that. Who is telling that? Because the viral load is less because of use of face mask, because the first inoculum of virus, that is will define your disease. If the viral load is less, then your symptom will be less, and it can be cleared by your immune system. So it will reduce your viral load and it will spreading will be lower. So use face mask religiously and wash your hand. And when you are outside, avoid touch the surfaces, because SARS-CoV-2 is virulent on and it can infect from the surfaces. It's 3 days, even 3 days it can it survived on the surface.
So another tricky question. In your presentation, you told that this viruses come from bat and mongolid. Simultaneously, you are telling bat is not our enemy. So how is it possible?
Bat is not our enemy because bat is a really integral part of our environment, and bat has the equal right like all other organisms have. And as I mentioned that we know bats are reservoir, but maybe some other reservoir also there. The all other rodents we don't know. And because we are encroaching their territory, they are nocturnal. There is a less possibility to contact with us. But because we encroach their land, that's why bat is coming. And from bat, it can be transmitted to other animal like Hendra. It was infected horse, from horse to human. So it is always it will happen. We have to protect the wildlife and the habitats for animals.
So many attendants are appreciated your presentation, very lucid
Thank
manner.
you. Oh, another mention that, as I told, that bats are a natural pest killer which can save millions of dollars for the pesticides and the harmful effect of the pesticides, and it required for a bat really help for the pollination of the fruits like banana and mangoes. So bat is really beneficial for us. Okay.
Next question, maybe technical, I don't know. Why is vaccine not made from polymerase genes? Because polymerase genes are less evolved with time.
Because we have to look at the antigenicity, and S protein has the highest antigenicity for this SARS-CoV-2. Scientists found that. That's why people are trying this S protein, because it is in the surface and it produces the highest level of antigenicity. And other structural protein, they also produce antigenicity, but it is less.
So is there any kind of infection peak and how it can be evaluated? The time period.
For asymptomatic, the virus shedding will be within as a median of 2 days, but for a symptomatic patient, it would be like 14 days, 21 days, and very critical patients, it can be prolonged for 1 month, the virus shedding.
Oh, is this overloading your question or I can continue? I don't
No,
know.
I am fine. I am fine. It's up to you.
How can I differentiate between fever and pollen allergy related respiratory symptoms and COVID-19 infection?
Though it's a, I think, question for clinicians, but I think that for allergy, you have only that breathing problem, but we don't have other symptoms like fever. I don't know personally, but I think so. And one interesting thing that today I read that actually who has the allergy, prone to allergy, they have the SU2, the expression of the SU2 receptor that is actually less in their body. So they are less prone for the virus infection. So maybe allergy prone people are safe a little bit. I can't conclude it, but I read it in a paper.
Okay, so sir, the death ratios are varying from one country to another. May I guess for the risk? May I guess for the racial variety of human host, or else the possible reason?
Uh, it can be told in two ways. Like if you see the death rate is in Europe is very high, like Italy and Spain, there's a like 10 more than 10%, 12%. Actually, their system is too overwhelmed. They didn't test all people. They only tested the severe symptomatic people. That's why it is not actually 10%. And for racial thing, yes, if you see the USA result, the more the death rate and hospitalization rate in the blacks, in the Hispanics, and the Asian, because they are poor, they don't have the medical insurances most of the time, and so they try to tolerate the disease as far they can. And when they are going to the hospital, then they are in the very severe stage. So that's why that economic thing is one of the associated with race type of thing. It's not that the any other physiological purpose. I don't think so. It is actually for the economic background. And because these, if you see the people here in USA, I can say that the black people, they have to come out for their work. They are janitors, they are drivers, so they are also, they can't maintain that much social distancing for their livelihood, so they are get infected and they don't have insurances. So that's the reason.
So nowadays, if we find little fever, cold, or throat sore, every tests are now. Should we test for COVID or go for medicines for fever?
uh, I think that it's a question for a clinician. I I don't know. Maybe you have some symptoms, and now because you have to stay home and to watch and contact your physician. Like here, it is also not possible. We have to video conference, and then we have to tell the symptoms, and then they recommended that we need a testing or not.
Next, maybe a dichotomy. There were some reports in newspapers regarding reverse zoonosis of the coronaviruses. Do these reports have any scientific basis?
I also heard that. I don't know, but it is reported that from reverse zoonosis means the dogs and cats, they are infected from human. They're actually their owners. But there's few reports. I don't think that's a very important thing. But people told me that reverse zoonosis also can happen.
Okay, maybe couple of questions can be taken, sir. Is there any chance to get reinfection with SARS-CoV-2 in an individual?
If we consider the first SARS, it was reported that until 2 years you have the protective IgG in your body, so you should be protected if the same thing happened for SARS-CoV-2. But still, we don't have any evidence for that. But personally, I believe theoretically you are protected, at least if it is not that much mutated, you are protected from the second infection. Otherwise, our vaccine and these herd immunity, all concept will be in vain. Dr. Vodro.
Yeah.
Yeah.
Where the coronavirus can be alive or not, cannot alive?
There is a long-term debate. The virus is either is a living material or non-living, but now there is a consensus that it is an intermediate virus. Means not for coronavirus, all viruses, they are intermediate between living and non-living, because they don't have any cell, they don't have any metabolism, they can't produce ATP, they can only multiply, but that multiplication also needs some host cell. They can't do it their own. They use the host machinery for their multiplication.
Can asymptomatic individuals infect others? WHO seems to be experiencing division of opinion about this.
Yeah, they confuse people. If it is in the first few days, like it is a median of their viral shedding is 2-3 days. In that time, they can infect, but you don't know when they are actually there. They have these viruses with them, because it's asymptomatic. Maybe today you are not shedding, tomorrow you will shed the viruses. So that is very difficult to say, to detect that who is asymptomatic and when he is shedding the viruses to the environment. That's why you need mask. It is not protect yourself, actually protect others.
So enough questions are being entertained. So we have come.
Dr. Dasgupta.
Yes, Dr. Salam.
I have one question, one or two questions.
Yes, please.
In case of higher education institute like college or universities, you know that there are canteen facilities are there and common bathroom facilities are there also. So how the students or faculty members should took the precautions and the sanitization frequencies are required for that particular situation in the higher education institute.
It is reported that although that SARS-CoV-2 can be our intestine, but there is no report that it can be transmitted through our stool. So that is one thing. And for personal hygiene, that if you touch something, you are carrying the virus and you are touching some surfaces, and again other people touch that surface, so it can happen that you are getting infected. So wash your hand as much as you can, and don't hand your mouth on.
Sanitization is required on frequent basis.
Sanitization, and sanitize all of surfaces. That is also required. Using the bleach. Bleach can destroy the virus particles. Using bleach, that will help.
And do you think that tree plantation reduces the chance of this infection of coronaviruses in the higher education institute?
What is the basis of that? I don't know. Why are you asking that? Tell me. It's an interesting question, though.
Tree plantation in the institute area.
No, I mean, how tree plantation is very good thing. I always encourage that. But why are you asking? Means, did you read something like that tree plantation can help?
I don't know. In another literature I was reading in a particular journal, there is some questions on that particular topic that tree plantation may reduces the chance of the spread of infection through this virus.
I am not sure about that, but yeah, I always encourage that concept, that plant as you can.
Another question: do you suggest vitamin C and zinc to fight?
Here, people are suggesting, and I can show. This is the vitamin C I am taking. I don't know, but it is reported that vitamin C can increase your lung function, and here doctor is recommending to take vitamin D and vitamin C. So I am also taking vitamin D and vitamin C. And for zinc, zinc is also known for other diseases, that if you administer zinc, it can induce boost your immune system. Though I have little doubt about that boosting immune system, because in our immunology books, I didn't read about that term, the boosting. Is it a commercial term or really scientific term? I have doubt. I don't know. But yeah, zinc used for other diseases too, and it has been reported for good clinical outcome.
So maybe the last question. Today there is an article in the newspaper that antibody of COVID-2 can be functional only for 2-3 months, and the same person can be affected after that. How far it is true?
I didn't see the report. If it's so, then it is really a problem, because for SARS-CoV-1, it was IgG was for 2 years, 720 days. If it is less than that, it is unfortunate. But I think that if we have infected once, it will generate our adaptive immune system, and it activate our B cell, T cells, so it can clear the virus. Because otherwise, why you should develop the vaccine? Same thing can happen for vaccines. The vaccine. So I think we should be hopeful for that, when not with that much pessimist.
Okay, let me introduce you to our Vice Principal, Dr. Sanjit Kumar Das.
Hello, Dr.
I would like
Thanks,
to Dr. Das to conclude the session with thanksgiving to the speaker, because we are.
Thank you, Dr. Dasgupta, for your lucid presentation and information shared with us. Hope we'll meet. We'll come here. And
yeah, sure. Veerabhumi is my Padar college, as I told you. Sir, your voice is broken. I couldn't hear.
Hello.
Now you are.
When you will come home, we'll meet. And apnar Padar college, apnar nemontorno roil.
Thank you.
Thank you very much. Thank you for your presentation.
And congratulations for organizing this beautiful webinar.
Thank you.
Okay.
So, Dr. Dashgupta, thank you very much once again for accepting our invitation.
Thank
So,
you
the
for
next
inviting me.
session is from your sir, our
Yeah, I am
own
eager.
sir, Dr. Dhruwajyoti Chattopadhyay. He will be joining us shortly. And I'm happy to introduce with you with sir.
Join. Please ask her to join.
হ্যাঁ আমার সাথে কথা হয়েছে স্যার জয়েন করছেন এক্ষুনি
Okay. Okay.
ও স্যার ছিলেন না আমার টকে ম্যাডাম স্যার ছিলেন
প্রথম দিকটা ছিলেন তারপরে বেরিয়ে গিয়েছিলেন So welcome you sir. Greetings from Bhairav Ganguly College.
Hello. Hello.
Namaskar.
Namaskar. Could you hear me?
Namaskar.
Could you hear
Sure.
me?
Yes,
Yes.
sir. Totally audible.
Okay.
So before we go into the introduction of Professor Chattopadhyay, may I now request our Vice Principal Dr. Sanjeev Kumar Das to welcome you on behalf of our college.
Sir, good morning and welcome to this today's webinar. And as a keynote speaker, we have concentrated us. That's our pleasure. And may I request you to continue deliver your keynote speech. And I'd like to request Deobrata to continue.
So it is indeed a pleasure to introduce Professor Chattopadhyay. Mr. Chattopadhyay is currently the Honorable Vice Chancellor of Sister Nivedita University. He graduated from Bachelor of Science from Presidency College and major in chemistry. He did his master's from biochemistry in 1975 and completed his PhD from Department of Biochemistry at Bose Institute. Professor Chattopadhyay was the Dean, Faculty of Science at the University of Calcutta from 2003 to 7 and was the Pro Vice Chancellor of the university from 2008 till 2015. He was also the director at the Center for Research in Nanoscience and Nanotechnology at the university during 2007 to 2015. He was the BC Guho Center for Genetic Engineering and Biotechnology and Department of Biotechnology and Biochemistry, University College of Science. He has received numerous awards, accolades, and distinctions in his career. He received the prestigious UNESCO IUMS MIRCENS SGM Fellowship at the University of Texas. He was also the Fellow of the Indian Academy of Sciences in 2004 and Fellow of the West Bengal Academy of Science and Technology in 2006. Dr. Chattopadhyay has lifetime members in various organizations and scientific associations. He has a teaching experience of more than 35 years and guided many PhD students. He has around 150 research papers and 6 book chapters in his credit in date. He has the H index of 34 and a very good citation index of above 3200. Professor Chattopadhyay is a scientist and academic par excellence. He has an established track record of highly innovative ideas and in-depth research activities. So, with this, I would like to request Professor Chattopadhyay to deliver his keynote. Over to you, sir.
Thank you, Dr. Bhadro, Dr. Das, Dr. Salim, and other participants over here, and definitely the IQAC. Very good opportunity for me to be present in a lecture which started with one of my very good student, Nirmaldo. And when Nirmaldo showed his introduction slide, I find what else I'm going to tell. So I said, Devabroto, what I'm going to say, because he's covering most of the things what I plan to say. So let me try and move a little bit out of what Nirmalo mentioned to you. So though I'm going to show my slides, I shall skip most of the slides. All right. Now, I really appreciate the people over here, the faculty members, and particularly the Department of Physics for, I shall request you when I shall go through the slides, okay? So that is one of the most important thing. Now, this is a picture of my first slide. You know, while the onset of COVID-19 has jolted the world vis-a-vis our country, as well as it has been a continuous process of protecting its citizens and ensuring that essential items are available seamlessly, learning, as we all know, can never stop. And this COVID-19 lockdown actually given us a lot of different other opportunities. And one of the most important opportunity I got it, that after a long period of time, I could be able to go through large number of articles, large number of scientific interactions. And all these things are possible because now we have the time to get and interact with many other people globally. So the whole world is now together. So any part of the world, we can actually connect and start discussing about it. If you look at this particular one, this particular first book, The Virus, it was a wonderful book. And I request all the participants over here, if you could get a chance, that if you can go through these books, The Virus, another book by Albert Marion, The Very, Very Dreadful, The Pandemic, and this particular book on Zika, Ebola, influenza, fatal fevers, more deadly war, and the vaccine race. All these particular books actually tells us that, you know, that what we are facing today, the world faced several times before. And really, 100 years back, when the Spanish flu was in our country, in the whole world, large number of people died globally. But at that time, one of the important difference from today's thing is this: that time, mostly the developing and the poor nations got affected. The wealthy nations are not affected in such a big way. Whereas this time, what we could find, the wealthy nations got affected in a major way. So this is a difference, actually, if you look at the type of infection, what we could see right now in the United States, in Europe, in most of the developed country, are actually exposed us that you may have a fantastic medical infrastructure, but still you cannot fight this minuscule virus particle. As mentioned by Nirmal already, the virus particle which we could not see under microscope. We need electron microscope to see it. And that is actually making all of us puzzled. So in that background, my presentation is this. And you know that in the middle, you can see a canvas ball. The canvas ball with the pins on it. So it is actually one of my students, PhD student's son, who is 8 years old. During this lockdown phase, at his home, he actually heard so much about the coronavirus, the corona coming out of that. On his canvas ball, he actually did it. So all these things are innovation. All these things are creativity, which are very, very important. Next slide, please. And thereby, next slide. Next slide. So this is actually a cumulative confirmatory case. And if you look at, as I mentioned to you, that Western world, United States, South America, and also if you look at the Europe, is much more, the incidence rate is much, much higher than if you look at what else, Africa, the other parts of our globe. So this is something which is different from the usual pandemics, what we could see with cholera, what we epidemic, cholera or malaria, TB and other diseases where this world gets affected, the other part not gets affected. Next slide. And if we look at from the next slide, please. Next slide. Next slide. Next slide. So you can see that if we look at the history of pandemics, that it goes on from this particular bubonic flu to the smallpox, then we have the flu, then we have this particular plague. So, Dr. Salam, it's okay? Yeah. So the plague, and over here you can see the death numbers and death toll also. At WHO for this coronavirus infection declared this particular term pandemic on March 11, 2020. So on March 11, 2020, we got this particular pandemic situation. Next slide. Next slide. Next slide. And if you look at this coronavirus, COVID-19 symptoms and others, already through everywhere you could come to know about it, but the primary symptoms are the fever, the runny nose, the dry cough, shortage of breath, fatigue, as well as a body ache. Now, today in the newspaper, you have seen that the pinkness in the eye is also something, and the coronavirus itself is associated with the conjunctivitis also earlier, but at that time, that is a different strain. And its transmission is terrible. The spread from person to person through sneezing or coughing. And the respiratory droplets, which contains a virus, can remain on surface even after the ill person is no longer there. Next slide. And in this particular case, when we looked at these common symptoms, there is loss of appetite also, loss of smell also. And in this particular severe diseases, we find that there is difficulty in walking, confusion, bluish face, and then coughing up with blood, the kidney failure, and there is a decrease in the white blood cells. And then it has got a white high fever. But in many of the cases, we could see that there are asymptomatic patients also. So the features of the infection could be clinical features, could be divided into three phases. One is the stage 1, the stage 2, and the stage 3. In the stage 1 is an early infection where the viral response phase. The stage 2 is a pulmonary level. The virus enters the lung and causing this particular pulmonary symptoms. And the third stage is the hyperinflammation phase where the people started its ultimate thing. But in general, what we could find that the mortality rate of this particular virus is much, much less than any other. Because here the mortality rate on an average is around 1%. But the contagious rate is very high. Now, how actually when it goes into the lung cells, next slide, yes, you are right. The lung cell, you can see the lung is made of the alveoli, and this particular alveoli has on its membrane actually has got number of receptors, what we call as a ACE2 receptor. And this ACE2 receptor recognizes the virus spikes, the spike protein on the virus surface, which protrudes out of the virus surface and which actually already discussed. Now, this particular case, if you start with the good alveoli, which is shown as first top, and then you can see there is infected one, you can see the virus particles inside. In the moderate one, there is fluid accumulation, and the severe one, it is completely filled up with fluid, and that's why the gas exchange, the balancing, the carbon dioxide oxygen exchange takes place there, and this is completely perturbed. Next. Next slide. And in general, what we could find that there are the organs which are affected. The major organ is the lungs, then the heart and the blood vessels, then comes the brain, the eyes, the nose, the various symptoms is we could see in certain patients, and that's because of this particular symptoms in the intestine also. So because these has got less number of ACE2 receptor, so here it is a secondary stage we can get this type of infection. Next. So virus actually enters the body through the nose, mouth, or eyes. And when it goes inside our body, what happens? It attaches to the cells in the airway that produce a protein called ACE2. And this ACE2 interaction, the virus enters into the body, fuses its membrane, and then endosomes. There is a change in the pH. You know, the acidic pH helps in the dissolution of the virus envelope, and the RNA comes out. It's an RNA virus, as mentioned by Nirmala, and this RNA gets translated into the cellular cytoplasm. So that is actually the case when the infection is in progress. And for each infected cell, it can release millions of copies of the virus before the cell finally breaks down and dies. So these millions of viruses coming out, then infect the neighboring cells, and it infects and goes on the infection. Now, in the meantime, what happens? Our immune system, it releases antigen presenting cells, or the APC, which helps to detect and identify the virus. And this ACP, this APC, antigen presenting cells, they engulf the virus and display the virus peptides, which allows other immune cells to recognize the virus too. So this is actually, I skip it because already mentioned by our Nirmala. But this is another thing many of us don't know, that this particular virus was first isolated as 29E by Dorothy Hamre, a virologist and infectious disease researchers at the University of Chicago in 1966. And June Almeida, working at Britain's laboratory, he actually took the first picture in 1967. Next slide. Next slide. Next. Next slide. So basically the virus which human corona. Next. Dr. Salam, next slide. So if we look at this particular virus, this is an important slide, though Nirmala mentioned about it, but I just want to tell you about this, that this Wuhan virus, its origin is from China. It has got its other contemporaries, the MERS virus, which is Middle East Respiratory Syndrome virus in Saudi Arabia, the SARS-1, also in southern China, and the caused by coronaviruses also. There are 15 to 20% of our common cold, actually it is due to the coronavirus. So all these other coronaviruses are not so dangerous like this Wuhan coronavirus regarding the transmission. And this transmission is so heavy. As I'm mentioning to you, the cells, APC presenting cells, they recognize the virus, and then this viral peptide is actually the one which helps us to protect ourselves from this virus infection. So as I mentioned to you regarding these 4 different types, you know, this Wuhan coronavirus is the one which is completely new and novel to us. So our immune cells are not ready to receive it. But if after APC presenting cells are being produced by our system, which was done by our immunity system, we can fight with it. But in many cases, if your immune system is not so strong, or due to aging or other diseases, you can have some, so that the system could not produce these APC presenting cells bearing antibodies, then they cannot fight with the virus. So I got infection with the virus. My APC cells, they are actually help the viral peptide presentation, and these T helper cells, they attach onto the viral peptides and being flagged by the APCs, and then activate the other immune cells called the B cells and the cytotoxic T cells. These B cells produce the antibodies that help prevent the virus from infecting cells, and the cytotoxic T cells, they identify and destroy these particular infected cells. The long-lived memory B and T cells that recognize the virus can patrol the body for months or years, providing immunity to us. And that is one of the reasons what we could find out that why we are actually getting asymptomatic also. So our immunity system fought with this initial virus infection, and we are not allowing the virus to replicate in our body. But still in our system, we can have a very low level of virus. So the asymptomatic patients, if the virus load goes off, they become symptomatic. A little bit moderate numbers, they will moderately symptomatic. So they may also give rise to this particular infection. But in general, the asymptomatic patients are due to our immune system. Now, this particular slide shows that how this ORFs, the open reading frames, they are actually helping this particular production of the replicase proteins. And these replicase proteins are actually helping the virus to replicate inside the cells. And that's why it is very important to know about the subgenomic messenger RNAs produced by this particular virus particle. And you know, this RNA is actually, this messenger RNA is actually the one which is being. Now, this slide actually shows you this infection process, the one which I just mentioned. Next slide. Next slide. No, next. Yes. Now, SARS-CoV-2 infection can spread faster. The reason for it spreading faster, it was found that this particular spike protein, the spike protein is actually, there is an enzyme called furin cells. The furin cuts the spike protein at S1, S2 site, and the spike protein then attached with the new host. In order to efficiently enter the cell, the spike protein still needs to be dependent on this enzyme TMPRSS2. This activation of TMPRSS2 is only possible if the spike protein has previously been actually been clipped by furin. So this is actually how I'm showing it, that what happens to this pre-activated one into the cell. So now if there is any way, this particular furin is present in various human organs, and because of this furin, where this particular cutting can be taken place, you can see this actually gives rise to the potential infection. But in certain genotypes of human population, we found that this particular TMPRS2 is actually very less. And if you have a less number of this, what we'll have, they are going to be resistant to cut it, and then there will be no infection further proceeded. So this may be one of the reasons. So immunity may be the first reason, the second reason of this, and the third reason what we could find, the ACE2 protein numbers, these particular receptor numbers may be lower in certain cases, for which another protein is involved, which we call as the ACE1. And if you have higher ACE1, you have less ACE2. In that way also it can control. Now, you know that the diagnosis is there. Nirmal already mentioned about it. There are several different ways of diagnosis are there. The real-time based RT-PCRs, these are all coming up in the newspapers also. and the other one is a rapid antibody test. But antibody will be there only to those particular people who has got its infection already combated with this particular virus inside the system and produces the antibodies. So unless the antibody, and in general it was found that the IgG and IgM, it takes a little bit time, 7-8 days and another 10 days to appear into our blood system, and only then it will be positive for us. So antibody test is actually going to help the already infected patients, but the newly infected patients we have to identify by this test. And thereby what we could find, that testing in India is pretty good right now. Large number of people are being tested. Joe, compared to the other parts of the world, the test number is less, but still we are improving on our testing facility, and every day it is coming up and up. But only concern to me is that the price of the testing, there should be a limitation. There are a number of scientists who came up with this particular thing. In our state, GCC Biotech already produced, and for testing purpose, I don't understand why it will take rupees 3,500. Now, it may be 1,000 or 1,500, but it goes up and up and up. So the major challenges for testing and diagnosis is the number of test kits, the quality of the test kits. You know, Delhi University, Delhi IIT also come up with a very nice test kit. IIGB, they have actually come up with a very interesting name, Feluda, as a test kit. All these are actually at the level of 500 rupees, but still they are not available at least. This is a Feluda, and these are based on this particular FNCase editor, the CASPACE edited linked uniform detection test. Next. So this number of tests, if it prices goes down, it will definitely help. So if we look at the treatment of this particular COVID, workflow, you can see this is a slide which explains the treatment and under which particular condition we have to get it. And also we could see that when it will give us an idea about the normal diagnostics and therapeutic, the negative PCR, the positive PCR for COVID, and the different types of admission to the different hospitals. Next slide, please. Next slide. So people are trying to make a cure of this particular. But over here, I just want to mention that Rabindranath Tagore, or in general, our grandmother and others, we already heard that it is mentioned that in case of the influenza or systems like that, ছাড়লেও 7 দিন, ওষুধ খেলে 7 দিন, না খেলে 1 সপ্তাহ. Similarly, Rabindranath চোখের বালি, in his through Bihari mentioned that it is better that instead of going to for any sort of medical person, if you can prevent it, it is much better. So that's why I said prevention is better than cure, which Rabindranath in Chokher Bali through Bihari mentioned already. So stay home, save lives, help stop coronavirus by doing these things. So the hand sanitizer, the wearing of the mask, and you can see that right now we are pretty aware of the fact, but still there are a number of people who are adamant and not wearing this. But you don't know that how many of them are asymptomatic. So please request everybody to wear the face mask and try actually to prevent this. And only in that way we can actually get out of this particular viral disorder. Next slide. Next slide. So in general, what we could find, the first human trial is actually done with the different vaccines, and there are different clinical phases. 3 is one of the candidate, AD5 INO4800, and the others are actually, as I mentioned, the large, the pathogen-specific APC, the antigen-presenting cells. Now, this LMP encapsulated mRNA vaccines encoding S protein by Moderna is actually very important. And I'll try actually to mention that here, the number of physicists are actually helping to prepare some sort of nanoparticles. These nanoparticles will help to encapsulate this mRNA for actually delivering the vaccine so that the vaccine is not going to destroy. Nanomaterials are also being used, as I mentioned to you before, that don't touch anybody. Why? Because the surfaces may contain the sputum or the cough or the sneeze of another person. You don't know about it. And that particular thing could be prevented. And there are number of nano paints are actually started to be available, which you could put it on the surfaces. They will not allow these virus particles to actually adhere to the surface. Recently from ISCS, Shudip Chatterjee and other group, Shudip Chatterjee is one of the research scholars over there, they actually reported about one of the material which is very interesting because of this electrostatic repulsion between the negative charges on the viral surface and the nanoparticles negative charges. They repel each other and not allow this particular virus to adhere to any surface. So there are these particular type of things are going on. But there is an adenovirus type 5 vector also. But all these things are actually based on the spike protein. There are DNA-derived vaccines, which also delivered by electroporation, where actually with the help of a difference in the voltage, it is possible to transfer the DNA inside the cell, and which codes for this S protein. And if you have in your body S protein already saturating the C2 receptor, then the new virus particle cannot get the scope. And it is very interesting that after this particular infection, within March, we could see the first crystal structure of this ACE2 S protein interaction. These particular crystal structures are available. This is a first in such case where in such a short time the crystal structures are available. So we could really thank the scientists, researchers for doing so much for this particular purpose. So in one hand we have the doctors, the nurse, who are actually helping us to prevent to get out of this particular disease. The scientists, researchers of different disciplines combining the work together, they're coming up with different processes, starting from preparation of ventilators to nanopens to the medicals to the encapsulation. It is possible actually to develop the new molecules, new drugs, how to deliver them. And the other one is definitely the police and other machinery, which are helping that people how to keep these people separate. I must thank the also the people of journalism and communication who are playing very important role during this period of time, informing the people about the different stages of this particular. Every day we are coming up with informations through them, which are not there. Now, so there are major vaccine projects over here. Next slide. Next slide is okay. Yeah, the Moderna vaccine, which we are expecting very soon. And this is actually one what is already mentioned and mentioned by the Nirmal. So I shall not go through it. Next slide. Next slide. Next slide, please. And over here, I just want to know, or I want to tell you that the other approaches what is being taken is a repurposing of existing drugs. You know, the drug development itself is a 10 years, 20 years project, and itself costs a huge amount, but we cannot wait. So what was the other try the people are doing is that there are a number of drugs to the virus particles and inhibiting their activities, and to see which are FDA approved already, so that we can use those drugs, modify them, and use for our own purpose against this. And in that way, there are several drug particles being identified, and you know that it started with one of the medicine we call hydroxychloroquine. This it actually increases the acidic pH of the endosomes. So if you remember that after the immediate infection, the endosome's acidification is involved in the RNA to get released. But if the acidification gets, the pH goes up, then this particular release is not going to be there, people thought. And they tried to have this medicine. Later on, we find that when it has been globally being applied and at a lower dose, because a higher dose, it has got some side effects, the efficacy is not so high. There is remdesivir also, which is coming up, but that only what happens, the result looks like that it delays the infection process by 30%. But there are many other RNA-dependent RNA polymerase inhibitors which are coming up right now. Flaviprovir as the one which is used as a flu, against the flu, was found to be very potential drug. In certain cases, prednisolone, the dexamethasone, and they are actually helping also for the terminal patients. So there are a number of drugs which we call it as a repurposing of existing drugs. Besides that, computational biology is helping in a very way to actually model this interaction and try to find out how to actually prevent this interaction so that the virus access to the receptors. Next slide. So the drug developments I mentioned to you. Next slide. Next slide. Next slide. This lopinavir and this hydroxychloroquine I mentioned to you. Next. Next slide. Over here, I just mentioned to you another new one, which just in the month of June came out, is that competing pair of human neutralizing antibodies, which actually been blocked the COVID-19 virus binding to its receptor. And there is another one, development of an inactivated vaccine candidate for SARS-CoV-2. These are, and in the Nature, another one came as cross-neutralization of SARS-CoV-2 by a human monoclonal antibody. All these three are very, very promising right now. So when we talk about the types of vaccines, it could be 4 vaccine or the inactivated vaccine, which is very successful in case of polio, the Sabin salts vaccine used for polio. The viral vector vaccines, it may be the replicating viral vector vaccine or maybe the non-replicating one. The nucleic acid vaccines, as I mentioned to you, it may be DNA RNA based, or the nucleic acid vaccines, which are protein based, and this protein subunit based or the virus-like particles. These vaccines are all actually right now under the test, and there are almost 75 very promising candidates out of 123 actually reported. But 75 was found to be very, very promising one. But one of this particular vaccine from Oxford, they said that they may be available to the people for the final phase in the month of end of September. The vaccine companies in India is playing very important role. The Serum Institute already are involved in the wicked vaccine program, the other programs also. The Oxford group also with AstraZeneca. Over here, the Bharat Biotech is ready to prepare that in bulk amount. So Indian vaccine companies are already preparing themselves that once the vaccine will come, they will manufacture it in large number so that the Indian people can get it. Next, at least the Indian people can get it. Next slide. Yeah. So as I mentioned to you, that these are adaptive immunity and the innate immunity both plays very important role. And this innate immunity is a word which makes one population very, very, what I could say, the strong against this particular viral war, but the one having this immunity not so high are going to succumb to this, and they have to take a lot of precaution. And the second thing, as I mentioned to you, the age of the, in general, the Southeast Asia population is much less than the other parts of the country. In our country, the number of people in the young age is very high. So that may be another reason why the infection is not so high. But I must congratulate that this particular lockdown at the initial stage, when our medical infrastructure is completely not ready with it, played a very important role to slow down the process of infection. Now the people are aware of it. The lockdown is slowly moved out to unlock, but we have to see that people are aware, they are maintaining the social, this physical distance, they are having the masks, they're taking the precautions with washing, with the we don't have to wait till a huge population, because there is another theory of herd immunity where at least 70% of the population, once it gets infected, it will be known as herd immunity, which help the infection to control itself, because virus itself is a non-living, as mentioned by Nirmal. It only gets into the living status when it gets a person or a cell, living cell. But if it could not get the living cell, it will not have any effect on that. So this is one of the reasons. But in India, if we have to look for herd immunity, I'm sure that will take care of 97 crores of our population at least to get this particular infection, which is very difficult for us to afford it. So adaptive immunity is a one which by which we have our production of the antibodies in our system. And there are also the interferons levels, which are also very important in this. But you know, this pro-inflammatory results produced by cytokines, and then ultimately when the inflammation, if you remember about the slide where in the third phase there is inflammation, there is a cytokine storm, and only if we reach that particular stage, there is no other alternative, but probability of death will be much, much higher. Next. So these are the different types. Action. I shall not go through it. This is a very technical one. Next slide. Next slide. And this outcome of this pathological inflammation in patients with COVID-19, as I mentioned to you, it is very difficult when the cytokine storms takes place, and the inflammation itself will be dangerous. Next slide. Next slide. So in that case, the immune suppressants has to be used. We have to decrease immune reactions, and immune suppressants can use it. And in certain cases, it was found that the blood thinners also helps. But in general, there is a hole in this plays important role, and now it is completely negated also. Next slide. Next slide. So in general, what I could find that the possible contribution of the hyperactivated monocytes to coagulate in COVID-19 is one of the reasons why the blood thinners sometimes help. Next slide. Next slide. This is an overall picture, what we have actually discussed so far in the disease management. And disease management involves all these things. And I request everybody, this information, knowledge, data integration is continuously going on. Every day we are coming with new knowledge, and so fast knowledge acquiring is only possible because this particular disease have been looked as a globalized disease where the whole globe is right now fighting with this virus, trying to come out. The other important things what I could find is a collaboration. There are a number of collaboration starts between different countries to actually fight for this virus. Third is the one where we understood that our mindset has to be changed. The mindset regarding what we learned so far, there is a possibility that we have to unlearn those things. And if we unlearn those things, we can relearn also. So all these things are going to be very, very important to all of us. Next slide, please. So at the end, I must say that COVID stigma is the one which is the worst thing over here. I came to know from number of people that how stigmatized are there, how panicked the people are in an area. And the very fast you can see the nurse who are working for us, the community is not allowing them to enter that particular area where they live. The other thing is that, oh, that particular person has got COVID-19. So how actually we could actually make completely that person itself. You have to be very understandable. Yes, we are isolating to prevent the infection, but we are going to help you. Don't worry. Your family. There are racial stigma, there are violence against health worker, and also there are social stigma. The affected individuals are facing social stigma. All these are very, very bad. So we have to fight it out. We have to be individuals. Right now we can see the other therapy, which we call as a plasma therapy, which is all where we are still trying to do that. There are so many COVID-19 cured patients. They're offering their plasma for the others. So we have to be kind to these affected individuals. We have to depend on the facts and not on the rumors. We have to spread awareness regarding do's and don'ts as endorsed by our health workers and government, and providing the mental health care, because the stress is another one which is going to affect in a huge way. So each one of us has to fight this stress and help others to fight that stress also. So all these things are, if we can carry on with it, because we don't know how long it will continue, maybe till the end of this year also, or it will slow down a little bit, but still we learned a lot from this COVID-19 pandemic, and we have to keep our learning with us, that this is a process of relearning. Next slide, which is my last slide, is actually where I wanted to make mention about this whole thing regarding the stay safe and try to actually help the others stay home, stop coronavirus, and stay positive. So huge thanks to all the participants, huge thanks to everybody and the COVID warriors who are working for it. Thank you very much.
Thank you, sir. Living in Calcutta, who else can be the better person to address the gathering as keynote address?
Thank you very much.
So, as you know, in the keynote session, there is no question answer, but sir has kindly consented if you allow for some questions to entertain. Maybe a silly one for you. Can I go, sir?
Yes, 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 definitely.
So the first question is, why we not use prokaryotic translation inhibitors against COVID-19?
You know that COVID-19 only after infecting the cells starts its activities. And what type of cells they're infecting? These are the eukaryotic cells. So in the eukaryotic cells, prokaryotic transcription inhibitors are definitely not going to work. Their machineries are completely different.
So a innumerable number of thanks to you from the attendants.
Thank
And
you. Devarbrata, what I can tell you that if you could find a couple of questions together, you can send the email to me. I shall send the reply, because I don't want to, because already you are late. And so I don't want to keep your time to be actually properly managed. I don't want to disrupt that. So send me the email. And Devarbrata, I really want to thank you and Dr. Salam for his help in my presentation of these slides. And thanks all the participants for the wonderful expression you have given.
sir, one thing I must mention, you have a very good library behind you.
Yeah, this is my library room. Yeah, home library.
So I remember Professor Chattopadhyay has visited our college a couple of years back during his convocation address in our
Yes.
college. So we thank you, and such a comprehensive lecture. I think there is any stones that are being untouched. So thank you very much, Professor Chattopadhyay. So for the participants, sir has allowed you to conclude that if selected number of questions are being sent to his email, he would consented to answer you back. So thank you
Absolutely.
very much. So thank
Thank
you, Dr. Salam.
Thank you. Thank you, Nirmal.
Thank you, sir.
Yeah, nice to see you. Nice to see you.
So the
Nice
session,
to meet you.
so the session of student-teacher duo is comes to an end. And for the next session of the participant, I request all the participants to log on to 3:30 when Shumita Goswami from Portugal will deliver her lecture on COVID-19. So that's all for this session. Thank you very much. Thank you for attending the.
With the help of the second YouTube link, you must mention that.
For the next session, you have to click the second YouTube session when the Zoom attendance as well as the YouTube sessions will be live right at 3:30 today. Thank you very much. Thank you all the panelists and all the participants. On behalf of Bhairav Ganguly College, we welcome you all once again, and thank you for the nice introduction by Dr. Nirmalo Dasgupta and Professor Dhrubajyoti Chattopadhyay. We thank you again, and we conclude the first session here. Thank you.
Thank you very much.
See you soon, sir.
Thank you.